Hey, Hammy here with the first video from Chapter 7 on which we're going to look at the process of photosynthesis. The different sections here of photosynthesis, and some of these are going to be combined. For example, in this first video I'm going to work through 7.1 photosynthetic organisms and 7.2 the process of photosynthesis, just kind of an overview and some history. And then 7.3 will be kind of the main, like over the first step of photosynthesis. 7.4 will be over the second step of photosynthesis. And then the last section are some kind of variations of photosynthesis that we find out there in the plant world. First, just kind of some background, and some of this will be review for you. Photosynthesis does take place in the green portions of the plant. And a lot of times that's what we call the mesophyll tissue. When we look at the leaf, kind of a cross-section of leaf here on a future slide here, I'll show you where that's at. So again, just like humans, plants would have organs and tissues, cells, tissues, and organs. So the leaf would be considered the organ, and the mesophyll would be considered the tissue where this is happening. Green because chloroplasts contain chlorophyll that are going to carry out the process of photosynthesis. The raw materials, remember the equation for photosynthesis. Okay, we've got 6 CO2 plus 6 H2Os in the presence of light from the sun, oftentimes are going to make a sugar, the form of glucose, C6H12O6, and give off 6 molecules of O2 oxygen. Where do some of these things come from? Okay, the water is going to be taken up by the roots. Okay, so it moves up through the vascular tissue. Water, in this case, moves through the plant through a tissue we call xylem, and you'll learn more about that in the plant chapters. Carbon dioxide, this material over here, is taken in through the leaf. Okay, so plants have to breathe, but not like we think of with a diaphragm and lungs and forcefully kind of, creating a vacuum and inhaling. They have openings called stomata in their leaves. I don't know. What's stomata with you? Sorry, had to slip a dad joke in there. Okay, these stomata are openings in the epidermis of the leaf that allows CO2 or carbon dioxide to diffuse into the leaf and provide the mesophyll cells with the raw material or the carbon source to make the sugar. Within the chloroplasts of the organelles where these are occurring, just talked about them, there are thylakoid membranes that contain the chlorophyll. Okay, the chlorophyll is what is going to make this organ green, or these tissues green. And there are other pigments as well that can help absorb solar energy, but chlorophyll is the main one we often talk about. The sunlight is used to energize electrons, and then the electrons are dumped into carbon dioxide. And if you remember from last chapter, that means that carbon dioxide is reduced, because we add electrons, which means CO2 is reduced to form a carbohydrate. This happens in the stroma. Again, we'll talk about structure here in a second. Combines with water to make our end product, a high energy organic molecule called a sugar or glucose. This diagram is just showing kind of an overview, some terms when we talk about energy flow. If you remember from last chapter, chemicals cycle, but energy flows one way through an ecosystem. Okay, always starting here with the sun, okay, to the green plants with photosynthesis. Okay, we call these autotrophs, making their own food. I remember trough as something an animal eats out of. So what are you eating out of? If it's auto, you're making your own food to put in the trough. Okay, so they're spitting out glucose and O2 as byproducts or as products, which are taken in and eaten by the animals. Now, don't be confused. The glucose is used by the plant as well for energy. on the O2. Some of that O2 will be used as by the plant for cellular respiration, okay, to give off chemical energy for cellular processes that it needs. The byproducts of that are water and CO2 are put back into the air, and it's then cycled again. So you can talk about the carbon cycle and the water cycle and those kind of things. So these, the things that are eating the autotrophs for energy that have to eat other things are called heterotrophs. And you can remember we can break those into things like herbivores, omnivores, things that only eat plants, things that it can eat both plants and other animals, or carnivores, the things that are the meat eaters, or they only eat other consumers. This is kind of a cross section of the leaf that I was referring to earlier. On the outside, you have kind of a waxy cuticle that helps protect the leaf. Underneath that, an upper and lower epidermis on both sides of the leaf. And then in here are our mesophyll. These ones that are arranged kind of nice and neat are often known as the palisade mesophyll. And these that are a little bit more random are known as the spongy mesophyll. And you can see the little green dots in there. They contain the chloroplast and where photosynthesis is going to be taking place. Okay, so star that one right there. Here we see a stoma, the opening, and on either side of the opening, there's going to be two guard cells that can open and close this opening. Okay, if we need to shut them, or the plant needs to shut them so that it doesn't lose too much water. Here's a leaf vein. So the blue is the phloem. That's where the sugars are going to be taken back down towards the roots and stored. The red is the xylem, where the water, xylem is where the water is going to be taken up. Phloem is what takes the sugars back down. Okay, so that's kind of the veins, the circulatory system of the plant. If we look at a chloroplast, okay, here's a cross section of a chloroplast. Some terms to be familiar with it. Notice it has an outer and an inner membrane we talked about in class, which can refer back to the endosymbiotic theory, that it was once engulfed. It was its own little prokaryote, and then it was engulfed and made into kind of a eukaryotic organelle. The stroba is the liquidy part in here, where the second stage or light independent reactions of photosynthesis are going to take place. And then these little pancakes right here are called thylakoids, the little flat pancakey membranes, and this is where you're going to find your chlorophyll, your green pigment, and where the light harnessing or light dependent reactions, the first step of photosynthesis, is going to take place. And then the whole stack of thylakoids, okay, is called a granum, or grana for plural. Kind of an overview of the process of photosynthesis, and I don't want to spend a lot of time on this because we're going to go through these two more in depth in the next couple sections. First off, the light reactions. This is the first step. They are the light capturing or energy capturing reactions where chlorophyll absorbs the light. It's going to energize electrons. There is an ETS or ETC, electron transport chain, that are going to move these electrons down. And we'll talk about chemiosmotic phosphorylation. So it will make some ATP. The electrons can be re-energized eventually and be picked up by NAD plus to make NADPH. Okay, these electrons and this energy then is taken to step 2, the Calvin cycle, where CO2, the electrons are put in the CO2 to reduce it into a carbohydrate. Okay? The reactions, again, use the energy and the electrons to make this. They're named after Melvin Calvin, who I'll talk about here in a second. And we're talking oxidation and reduction. Okay, So whereas if you go back to the formula, and I'll just very quickly simplify it, CO2 plus H2O, presence of light makes C6H12O6 plus O6. Okay, remember, CO2 is going to be reduction, is reduced into a sugar, whereas water is going to accept those electrons, and water is going to be oxidized into O2. Okay, so there you have your oxidation and reduction happening here, what we referred to last chapter as a redox reaction. Okay, sidetrack here a little bit. I want to talk about the discovery. Okay, this is Hammy's adding into the chapter a little bit. I always like kind of doing a little bit of a history lesson because you kind of learn about how did we figure all of this out. It wasn't just one person that was like, boom, that's photosynthesis. It was a collection of scientists that thought about this. It started early on. We often give von Helmont credit for getting some of the thinking started. Most people say, where do you put plants? In the soil. Okay, you put plants in the soil, they suck up the dirt and the water, and that makes the plant. Well, he did an experiment for 5 years with a willow tree. And he started with 5-pound sapling and 200 pounds of dirt, okay, and planted it in a pot. And then he watered it for 5 years. After 5 years, that plant's almost 170 pounds. And then he's like, whoa, there must not be much dirt left. Measured the dirt, 199 pounds, 14 ounces. Okay, it only lost like 2 ounces of dirt. So how did 2 ounces of dirt become another 164 pounds of tree? Okay, how does that happen? Well, he concluded that it had to have come mostly from the water that he was adding, okay, every so often, because it really didn't lose that much soil. In the 1770s, Joseph Priestley made a discovery that he put this jar over a mouse, okay, and the candle would use up all the oxygen, okay, and so what would happen to the mouse? The mouse would be dead. But he noticed that if he put a mint plant in there, okay, the mint plant would keep the mouse alive. So he said, well, the plant must be giving off oxygen, and that's keeping, yay, mouse is happy. It's keeping the mouse happy. Okay. Soon after that, Jan Ingenhousz noticed that it plants only, he kind of repeated this experiment, but he noticed it only happened when they were in the presence of light. Okay, and it also was just the green parts of the plant. You can notice some aquatic plants will actually give off bubbles of oxygen underneath the water, and you can collect that. And so he repeated this where some of them in the dark, okay, some of them in the dark, and then some of them in the light, and only the ones in the light did the mouse stay alive because the plant was actively photosynthesizing. In the 1840s, Julius Robert Mayer made the proposal that, hey, plants must take that light energy and convert them into a sugar or some kind of chemical energy, because he noticed a lot of things eat plants to stay alive, and it's kind of the beginning of the food chains, right? And plants use a lot of it, need light to survive. So he said, well, maybe the plants are somehow taking that energy from the lights and turning it into chemical energy. Okay. In the 1930s, this progressed through several experiments. C.B. Van Niel is often credited for what very close to what we would call the final equation for photosynthesis, saying that, hey, plants take in carbon dioxide and water in the presence of light and then use that to make the chemical energy that Mayer was talking about in glucose, and giving off oxygen, okay, that the scientists before him were saying, hey, plants give off oxygen and keep the mouse alive. Okay, and he came up with this formula here. Then it really began to be worked on by biochemists. Melvin Calvin, who the Calvin cycle is named after, in 1946 began experimenting and using C14 a radioactive carbon to trace the path of CO2 or carbon through the pathways of being reduced from CO2 into a glucose. Okay, so here's CO2 being added into the pathway, and then out here's a G3P coming out, which combines with another G3P you'll learn about to make a glucose, and then RUBP is kind of recycled for this process to happen again. Okay, so he is really credited with figuring this out, and so it's named after him. Finally, here's just kind of an overview. This is important. I would star this figure in your book. This is important just as a basic understanding of the steps of photosynthesis. This is showing our inputs: light, water, carbon dioxide. It's showing the two steps of photosynthesis. Okay, first the light reactions, and then number two, the Calvin cycle reactions. You can see that the Calvin reactions are taking in light, they're taking in water, okay, and they're going to be giving off electrons, which are captured by NADPH, and some energy from the electron transport chain as ATP, which are carried over. That energy and electrons are carried over to the Calvin cycle, which turns that CO2 into a carbohydrate. Okay, these then come back and pick up more electrons, more energy, and they keep cycling between the two different reactions. These are taking place over here in the thylakoids, whereas this is taking place out here in the stroma. And finally, the end result, oxygen, is being given off as a product down here. So you have your inputs up here, and reactants and the products down here. So make sure you're very familiar with this. So that kind of sums up the overview and the history of photosynthesis. In the next videos, we'll start to get into the actual processes of photosynthesis.